There we go.
Okay, that work. You might need to mute yours. Oh, maybe. Oh, that's okay. Okay, so given these myriad costs and multiple fronts and trying to defend against multiple parasites at the same time, one expectation, right, is that we should see a lot of natural variation in some of these immune parameters. And so we can try to define what these parameters might be. We could have differences in constitutive production of some of these immune molecules, right? Even before you get infected, maybe you're just making more antimicrobial peptides or more phagocytes or something like that. You could have an increased maximum, right? Maybe there, some individuals have been selected, or some genotypes have been selected to make stronger or like very high immune responses, which would pay the cost of all that energy and immunopathology, but you might be able to kill off microbes. You could be more sensitive, right? So it takes fewer microbes to turn on an immune response. And so this could be helpful against a really kind of nasty pathogen that's growing really quickly, but it can also subject you to responding inappropriately to gut microbes or something like that in some of your commensals. And you can also just have what we call a twitchier immune response, right? Where it's kind of an all or nothing thing versus something that's a little bit more gradually regulated. All right, and so to try to actually test how variable the dynamics of immune responses are in wild populations, I mentioned that we could go out to these feed. This is a feed store. They sell food for horses and cattle and dogs and whatever animal you have. And you probably can't see them, but there's little beetles, right? They're all over this feed store. It's really hard to keep out all the stored grain pests. We can set these pheromone-baited traps, and these pheromones are relatively specific for our beetle species and one other beetle species that we often catch in these traps. Here's just a picture of me and one of my undergrads at one of the grain elevators that we visited. You can see kind of the grain that's kind of all around here, right? And this unrestrained grain can also attract pests. All right, and so this is work that my undergrad Abby Perry and my former RA Derek Gent did, where they took 4 wild populations of Tribolium castaneum that we collected, and 3 related populations of a related species, Tribolium confusum. And we infected them with 4 varying doses of our model pathogen, Bacillus thuringiensis, or a different pathogen, Photorhabdus luminescens. This one associates with pathogenic nematodes, and it's basically like the bioweapon the nematodes use to suppress the insect immune response when the nematode infects. And so we did these 4 different doses to try to get a variance in the starting density of our bacteria, so we could actually look at the kind of the quantitative variation of these responses. And we were measuring survival. Some of these pathway-specific parameters, like the constitutive production or the sensitivity, as well as overall resistance. So are they better or worse able to kill off the infections? All right, so first of all, these are 6 of the 7 populations that we assayed. One of them was so resistant that we thought we had screwed it up at first, so it's not on here. But you can see that among these different populations, right, this is hours post-infection with BT, this is proportion surviving. You can see that there's a lot of variance in these populations as to how susceptible they are to this particular infection. And when we mapped the BT density at 8 hours post-infection, these are the different populations. The different shapes or dose, but you don't really have to stare at that. But I just want to point out, right, that first of all, there's a lot of variance in resistance within populations. Part of this is down to these are all the doses kind of conglomerated together. But across populations, right, some of our populations are a lot more resistant to infection than some of the other populations.
So wait, then, can you go into a little bit more detail there?
About this?
Yeah, so first of all, I know that you inject these bacteria.
Yes, we inject these bacteria. So they grow really quickly. So they have a doubling time within the host of about 35 to 40 minutes, right? And so they're going to grow really exponentially, really quickly. And what we see is that usually within the first 12 to 14 hours, if these beetles are going to die, they die in that time, right? So this is a really rapid, thanks to the septic infection, it's a really rapid infection course.
And then on the right, so each symbol there is a population, not a symbol.
Each symbol is an individual, and each one of these is the population. Yeah. And so we kind of.
SNTS, and that's a population.
Yeah, so this is Marshall confusum, for example. This is the confusum beetles we collected from the Marshall site.
So therefore, each experiment, so these are not easy experiments.
No.
These are small, very small. And
yes, but we get pretty good at, like, you're using a stereo microscope and you have a little needle Oiku probably remembers, yeah. Yeah, it's hard at first.
I see them sometimes.
Uh-huh.
Like a millimeter. How big
Yeah,
is it?
tiny little needle, I think, I don't
No,
remember
no,
the
no,
size.
it's the animal, how big is it?
Oh, it's like that. But they're pretty big under the microscope, I don't know. The mosquito people are laughing because the mosquitoes in many cases are even harder to manipulate, I think. Yeah, it's one of those things where we have rotation students come into the lab and they're like, oh my God, how am I ever gonna get this needle into this beetle? But then we have stabilizing things, and sometimes we'll chill the insect to make it calm down so it's not flapping everywhere. And it's not too bad.
Okay, so the variability to the experiment is pretty precise, the variability cannot depend, I mean, that's.
Well, so part of this variability, if I go back to the idea here, right, we're sampling somewhere in here. And so we're already starting to see some of the bifurcation, and this is contributing to some of that variability. So if we sampled back here, we're going to see less variability, but we also don't necessarily have enough precision at very low microbe densities to get at variation among populations. So we kind of have to wait until we're up here, but then we're capturing some of those kind of trajectory variants.
So it's not a technical thing.
It's not, it's not really a technical thing. It's reflecting the variance here.
Okay.
Yeah. Some of it's a little technical.
For sure.
Okay, and so if we have all this variance, which is kind of hard to analyze statistically because it's so variable. But if we actually plot our antimicrobial peptide expression by the density of the microbes in these hosts. Like for example, if you look at this pink population here, this is our Green River confusum population. There's a really strong correlation between microbe density and antimicrobial peptide density. This is probably just related to the way the signaling works, right?
How do you measure that? I'm sorry.
This is via RT-qPCR. So we smash up the beetles.
Take the whole beetle.
Take the whole beetle and smash it. But you can also do guts. We've done different tissues and things like that as well. It shows some of these, so that the IMD regulated antimicrobial peptides tend to be super correlated to microbe density. The toll mediated ones and some of the other pathways aren't necessarily. So they have different types of regulatory dynamics. With the atacin one, for example, is under IMD control. And so you can see that there is strong correlations, but for different populations, they maybe have different slopes or different intercepts, right? And so if we think back to some of the parameters that we were interested in, like that initial investment in immunity, or like the kind of the threshold-like behavior, the kind of the sensitivity of these curves to microdensity, you can see we have a fair amount of variation among our populations. It's messy, right? These are wild populations. They're not inbred. There's a lot of kind of natural genetic variants as well that we're dealing with, but we can start to get some idea of what's going on. Right. And so this is kind of a messy plot, but basically what this is we measured a bunch of different things, a bunch of different parameters for these curves, like the intercept and the slope, et cetera. And basically, this is just a correlation plot. And we also have the hazard ratio. So that survival curve I showed you, we can get a hazard ratio from that relative to kind of our median population for both BT and our Photorhabdus luminescens, the other bacterium I talked about. And for example, we can see that for Photorhabdus luminescens hazard ratio, we have a very strong negative correlation with the intercept of some of our antimicrobial peptides, right? And if we plot this out, so here's, for example, defensin 1 intercept versus the hazard ratio. And so these are relative numbers. This is relative to a housekeeping gene. But basically what we can see is that the more defensin you're making, the lower the probability of you dying from infection, right? So investing more upfront in immune response does protect you against mortality. So you can see a really strong correlation. Not true for BT, right? Your investment in immunity has nothing to do with whether you survive BT infection. But what we found, interestingly, was that there's actually a positive correlation between the slope, or the kind of the twitchiness of that response, and whether or not you're going to die. So if you are twitchier, right, you're turning on everything maximally right away, as soon as you get infected, or not as soon as you get infected, but as you increase the microbe load, you're just trying to blitz it with everything you've got. That actually predicts mortality, right? You're more likely to die. And you think this comes down to the pathology of the immune response. A lot of these pathways, they don't just control antimicrobial peptides, they also control metabolism and organ homeostasis and things like that. We think these guys are basically just going berserk. That sure, they're trying to fight off infections, but they're actually causing more damage to cells.
Are all your beetles age matched whenever you?
Yes, yep, yeah, age and stage match, yeah. So these are, I believe these were size matched 14-day-old larvae. All right, and so we can make some initial conclusions from this work, where we see that an increased baseline is associated with better survival against photoreactive luminescence, right? Which is kind of what you'd expect in many cases. If you're making more immune response to begin with, and you have a better kind of barrier defense or whatever, you're going to be more resistant to infection. But interestingly, we also see this, right? Like, as you get twitchier, as you increase your slope, or you kind of increase the threshold behavior of this relationship, you get worse survival, right? We don't know why yet. So, you know, we don't think it's just because they're producing more antimicrobial peptides, even though that's our readout, right? That's the readout we're using. It doesn't necessarily imply causation. And so we're going back and looking at things like metabolic rate, the turnover of lipids and things like that to see if they're just burning through their energy too quickly. Are they destroying their gut in the process of trying to defend against infections? All right. So once you turn on an immune response, you also have to turn it off, right? Because these things are so expensive, because they're so damaging, you can't keep leaving it on. And so we can imagine the concept. First of all, if you think about one of our signaling pathways here, right, we can cartoon it where you can get different regulatory elements at different parts of the pathway, right? So, for example, if this is a negative regulator of a transcription factor, maybe it degrades the transcription factor, maybe it keeps it in the cytoplasm so it can't move into the nucleus, right? During an immune response, you degrade it so the transcription factor can start transcribing immune genes. We have things that are post-transcriptional, right? So they're turning off, or they're keeping zymogens inactive or something like that, right? These are regulators. And then we have things that can actually degrade the signal itself. So one example from insects is some peptidoglycan recognition proteins actually have amidase activity, so they can degrade peptidoglycan, so it can't keep activating immune responses. And so this is another way in which you can kind of regulate the strength of the signal going through that infection. And so if we think about the dynamics of turning off an immune response, right? We have a black curve here, which is if you're, as soon as you turn on, you turn it back off, right? And so you have this kind of fast decay rate in your immune response. Conversely, you could have a slow decay rate, right? That's in the light gray. The consequences of this, right? If you take a long time to turn off your immune response, you have more time to clear the pathogen. Whereas if you turn it off really quickly, you may not kill every reservoir of pathogen in the host, right? And so you can develop a chronic infection, right? So one of the consequences, for example, being too quick on the draw with turning it off is that you may not kill everybody, which may or may not be a problem, right? If this is a gut microbe gone wrong, you kill off the ones that are actually, in the outside the gut, and you leave around the ones that are in the gut. It's not an issue, right? And there are other examples of that, like a lot of insects have symbionts that they have a little bacterium for. As long as your symbionts are sequestered properly, it's not a problem. But then the kind of the trade-offs and the amount of damage that you ultimately accumulate from these different strategies is going to depend on the relative immunopathology relative to the microbe-mediated pathology, right? And so if here's, just our fast curve here in black, if the immunopathology is greater than the microbial pathology and you're slow to turn it off, right, you're actually going to accumulate more damage, even though you're clearing the infection at a greater rate. If, on the other hand, the microbial pathology is greater than the immunopathology, which we think about for most infections, right? Like if you have a really virulent pathogen, that's probably going to be true, but not always. Then you serve yourself better by turning off the immune response more slowly. Does that make sense? All right, and so we can actually manipulate this in our beetles. And so, for example, we can target, if we think about, we're thinking about this type of negative regulator here. In insects, the one that regulates our dorsal diff transcription factors and our toll pathway is called cactus. And we can actually we inject double-stranded RNA into our beetles. RNAi is super easy in our beetle system because all you have to do is inject double-stranded RNA and it goes systemic. And you can do this in any life stage. It lasts for anywhere between 2 weeks and several months, depending on the particular target, just kind of the random dynamics of it. We can achieve high efficiency knockdowns. Like most of our knockdowns are 90% or more efficient. So for example, here we have hours post injection with a heat-killed microbe. Okay, so we're trying to take microbial pathology out of the equation and just look at the immune response. Here we have the magnitude of an immune response, and this is RT-qPCR in one of our antimicrobial peptides. And so our normal course of action, right, is that this is the constitutive production of this antimicrobial peptide in a normal adult beetle. They are exposed to infection and they turn this thing on. So this is a log scale, right? So this is like 10 to the 5th induction of this antimicrobial peptide. And you can see that, it's really dramatic. It happens within 2 hours, right? So this is a really rapid induction, and it stays on for a while, even though these are heat-killed microbes, right? They're not reproducing, they're not producing more, maybe the breakdown products, the peptidoglycan, et cetera, keeps activating the immune response. But ultimately, right, the host is going to clear those antigens, and it's going to turn off its immune response, right? So we can see this really nice curve of turning the immune response basically back down to its pre-exposure levels. So what happens when we get rid of this negative regulator? Well, first of all, you can see that it's just turn on these things constitutively, right? It's incredibly strong induction of the toll pathway. Interestingly, we don't see any difference in the maximum, right? If we think back to our expectation for the different parameters, increasing the maximum was one of them, but we're basically already producing the maximum amount of these antimicrobial peptides. And they do start to turn the immune system off, right? After the elicitor is cleared. It actually gets a little bit lower than the constitutive levels, right? But it's declining at a slower rate than the beetles that aren't manipulated. Anybody have any questions about this? And we can do this for all different, we've done this for heat-killed bacteria, Bacillus thuringiensis, heat-killed Serratia marcescens, heat-killed Candida albicans. So we've tried a bunch of different pathogens to try to engage the different pathways, just so we can say, is this generalizable? And it is. They all are actually very similar to this. Yeah, Lindsay.
For your time point between 0 and 2 hours, measured
Between 0 and 2?
Yeah, if there's like a gradual increase.
I mean, does it go like this, or does it kind of go
Yeah.
like that? We don't have that for this time series. We're doing another time series right now where we're looking at translation efficiency between 0 and 2 hours, so we will have that data, but I don't have it yet. But I think it just, right, I think it probably just goes up pretty quick. I don't know if we'd actually have the resolution to figure out the functional form of that. All right, so this incredible increase in immune induction does translate into resistance against bacteria. So the cactus knockdowns do have lower bacterial loads at 8 hours post-infection, for example, and they're much more likely to survive lethal infections. So now we're infecting with live Bacillus thuringiensis. Those that receive the MALD control, right, this isn't targeting anything inside the host, it's just activating the RNAi pathway non-specifically, have about 50% mortality. We gave them different doses actually, so this is an intermediate cactus dose that achieves like a 60 or 70% knockdown efficiency, and here we're at 90% plus knockdown efficiency, right? So there is a quantitative variation that we can achieve in this system using different doses. But you can see that they're much more likely to survive infection, right? All right. But this comes at a cost to fitness. And so this was hours post-infection, right? I told you that if they're going to die, they die in the first 10 to 12 hours. Now we're looking at days post-dsRNA injection. Okay, so it's a different time scale here. The control individuals, these beetles will survive for months if you let them. They don't die very quickly. They'll run around happily for as long as you want them to. And so in that control dsRNA dose, they're basically all alive. But as we increase the dose of cactus dsRNA, you can see that we achieve stronger and stronger mortality effects. And this is in the absence of any kind of infection, right? This is just due to the knockdown of this negative regulator, right? So clearly, having constitutive activation of this cactus or this toll pathway is incredibly costly to the host, right? And they're dying in 6 to 8 days post dsRNA injection. And even before that, they get kind of stiff. We can tell there's something weird going on. They start to get listless, and then they keel over. But in this interval, so before they start to die, we measure their fecundity, so how many eggs they were producing. Here's our controls. These are given 250 nanograms of MALD dsRNA, so it's just a control for the RNAi pathway. And this is like the baseline fecundity of an insect. So in 3 days, they're producing 10 to 15 eggs, right? They live a long time, right? So if you actually do the math, they can produce hundreds and hundreds of eggs over their lifetime. But when we have this RNAi treatment, right, when we're knocking down cactus right away, even when we're only achieving 50 or 60% efficiency, you can see that their fecundity is really suffering, right? As we further increase the knockdown of that pathway, they're basically producing no eggs at all at this highest dose, right? So this is this constitutive activation of this pathway is incredibly costly to this fitness. And so this captures in many ways, right? It's the negative regulator of the transcription factor. It's basically like the bluntest instrument you can use to look at some of this pathology. But we want to go back and like look at some of the other negative regulators in the system, thinking about the different aspects of negative regulation, and trying to get at some of the kind of the costs and benefits of knocking down or manipulating the regulation at different points. And so what we can do, for example, is this is just 3 equations out of 10, but we have this ODE system of signaling pathway induction and decay, where we can actually model the kinetics of induction and decay. We can look, for example, at parameters that represent negative regulation. We can then like correlate that to the particular proteins in our signaling pathway and knock them down, right? So we can mess with these parameters to look at how we can then put the output of this, for example, the pathogen load over time, the area under the curve for the immune response over time, right? We can input that into a fitness function, right? And say, given kind of the calculus that's going on over here, how does that affect our predictions for your net fitness costs and benefits under different parasite conditions? And then we can actually go back to our natural populations or to our lab manipulative experiments and actually test the fitness costs and benefits of variation and regulation, right? We know we have a lot of natural variation in this regulation. We're looking for even more of it. And I think it's going to be really fun to try to correlate this to fitness and try to say, like, what is an optimal immune response in various conditions? Anybody have any questions about that?
Do you ever find outliers?
At the population level or the individual?
Like individuals that are outliers?
Well, it's hard. We do see a lot of variance from individual to individual depending. So I will say with the larvae, one of the things that we really struggle with, right, is that there's a lot of things that are kind of under the simultaneous control. Like if you have, for example, some of these larval hormones that correlate molting or something like that, one of the big contributors to variation in larvae in our phenotypes is whether they're molting or not. Because when they molt, they shed their cuticle. It takes part of the rebuilding a cuticle is using part of the immune system, for example, and so we see kind of these kind of induction, these oscillating dynamics with the molting. So that's a big contributor to variation. Males and females, I didn't show you, but a lot of this stuff we're doing is sex specific, and we have to make it sex specific because there is a signal of sex in the
Females.
These are all females. Yeah, we decided to go with females for now, but we have male and female data for a lot of this too. Yeah, fecundities, this is all females. We haven't tried the males yet.
So in humans, the immune system is simply developed into more adulthood. I was wondering, is there differences in the activity of the immune system in these beetles from
Different
the
life stages.
life stage you do it at? And do you think there might be a difference if you were to do this, for example, dose control in the adult animal, in the more adult animals, as opposed to like the 14-day larvae state that you do?
Yeah, so I should say this is all this, the second half of this, the trying to correlate the induction and decay to fitness is in adults, because when we knock down cactus and larvae, they die in 3 days because they need that pathway for development. And so I had a bunch of slides in here on stage specific immune responses, and I took it out for time, but they do. So the larvae and adults do show different responses in different life stages, and they show different sensitivity to microbe density as well. So the adults are twitchier than the larvae. Whether this, one of the things that we're really interested in, I'm actually writing a grant proposal right now to the NSF, is to try to understand how stage specific responses, and do you have a different optimum in a larval response versus an adult response, right? We think you probably do. Because you're facing different parasites, you have different trade-offs with growth and development versus reproduction and survival, right? And so you're allocating energy to different processes. And as I'm going to talk about in just a minute, we also think there's a lot of constraint in some of the earlier stages that's going to drive how much of an immune response you can make.
Can you, those equations for induction and decay, are those general to all proteins or RNA transcripts, or are you?
I mean, you could basically, if you had a pathway, so like, for example, it's like the P is the parasite, and it's, or I think it's the recognition protein actually, and so it's inactivated or activated. And then we can, like, for example, we have MIT88, and we have our protein kinase cascade, or the MAP kinase cascade, or whatever. We have activated and activated. So we have an equation for each one of these compartments.
So those are general
Yeah,
equations.
you could write this for
referring
any
that,
signaling
that's
pathway.
right.
Yeah, we parameterized it based on the IND pathway. You could write it for any pathway. So thinking about development, right? Does development constrain adaptive evolution of immune genes? And now we're thinking more kind of in terms of this ontogeny, right? Moving from larva to pupa to an adult. And it turns out that development does constrain the deployment of an immune response. And one example of this is Drosophila melanogaster larva. This little black thing here is a parasitoid egg that's been successfully melanized. And what the insect does is it creates like black sticky substance around the egg. And this host will survive, okay? It's defended against this parasitoid egg. And this was a third instar larva, so we're over here. But if this guy had been infected in the first or second instar, there's no way he would have survived. And the reason is the hematopoietic organ doesn't even mature fully until the third instar, right? So these guys are basically undefended against parasitoid eggs immunologically. At the same time, I talked about this molting, right? Where we have this, you're shedding your cuticle and then you have to remake it. And so the phenyl oxidase, which is one of the enzymes that's responsible for making the melanization, oscillates from low to high between each molt. And part of that is because some of these precursors have to be shuttled to tanning the cuticle, which leaves less to be allocated to melanizing parasites. And so when you actually, for example, infect these individuals at different points during this molting process, it correlates. They're more likely to die of fungal infection in the intermolt process because they have less phenyl oxidase to fill off the fungus. So we have this idea of pleiotropy, right, where we have a shared genetic architecture. This can be at the allocation level, it can be at the genetic level, where we're trying simultaneously to use the same product or the same gene, right, in pathogen defense and development. Since both of these things are contributing to fitness, you can imagine if these were correlated, right, like if you were always getting infected with a particular parasite as an adult, it would be nice to have a syndrome of sorts, right, where you're turning on the immune response as soon as you think developmentally that you're going to get infected. So this could be a positive thing. But there's also the sense, right, where if you're using the same gene product for multiple things and they aren't positively co-varying in their effects on fitness, you can actually have a trade-off or a constraint, right? Something that's antagonistic between the two traits. And so if we actually look at the different immune signaling pathways, like the phenyl oxidase pathway, the toll pathway, et cetera, all of these have an immune function, but many of them also have a developmental function, like neurotransmitter production, developmental patterning and hematopoiesis, insulin signaling and metabolism, embryonic development, cell proliferation, stress response, et cetera, right? A lot of these pathways are doing double duty in many different types of traits. And so our answer is that pleiotropy is potentially ubiquitous in immune systems, at least in insects, and we think this is probably true in humans as well. People have done kind of surveys of potential pleiotropy in humans, and one of the processes that comes out as being most pleiotropic is genes involved in immunity. So it's kind of an interesting thing to think about. But the problem is that, we've seen that developmental pleiotropy can constrain the deployment of immune responses, right? When in development, we can actually use these. But our question now is, can they also constrain their evolution? And the reason we think about this, right, if you think about the strength of selection on developmental genes, right? When you're an embryo, these things are under incredible purifying selection, right? You're trying to keep this module as is, because if you mess with it in any way, you're growing an extra limb, or you're not developing at all, right? It's incredibly hard to mess with a really intensely co-connected set of proteins during the embryonic stage. But as you go through development, right, like adulthood, you survive the juvenile stage, you have a little bit more variation that you can mess with without completely killing off the fitness of the insect, right? And so the strength of purifying selection on genes that are involved in juvenile or adult development are probably a little bit less constrained than those involved in adult development.
So then.
Yeah.
There's a couple of terms that you need to explain.
Okay.
So I know for my peers that some of these terms are not necessarily common jargon across biology. I was out. Did you explain what pleiotropy is?
Yes, it was
You explained. Say one gene.
one gene, multiple traits. Yeah.
Multiple traits. And so what about the purifying?
Yeah. So purifying selection. So if you think about the way selection works, right? You can have let's say you have a protein, and you change a nucleotide such that it changes the amino acid it codes for. All right. Now we're encoding a different protein, right? A different polypeptide that may fold differently and may interact differently with its interacting partners, right? And so the question is, are you allowed to maintain this mutation, right, that changes the function of the protein? If the answer is no, right, it's changing the function of the protein such that it hurts your fitness, then we expect purifying selection to act. We're going to get rid of anybody or any genotype that has that mutation because it's detrimental to fitness. That's purifying selection. On the other hand, if that mutation actually ends up being beneficial, you may want to select for it, right? In which case we might expect to see positive selection. We're actually taking this new innovation and it's going to rise in frequency in the population because it's beneficial in some way. Okay. And so if we think about the proportion of these adaptive substitutions, for example, this is a statistic called alpha. These are different arms of the immune system in insects, and you can see there's a lot of variation in the strength of selection on these different proteins. So for example, cellular and encapsulation processes have a high proportion of adaptive substitutions. Some of these, on the other hand, have a very low proportion of adaptive substitutions. Some of this is related to the strength of selection, so how efficiently you get rid of things that are deleterious. And so you may see a high proportion of adaptive substitutions, for example, if selection is very strong, because you're going to efficiently get rid of anything that's deleterious and efficiently keep anything that is beneficial.
Are all these being evaluated in a single environment? Because
This is at the genome level, so you're looking at the, I'm going to explain it actually in the next slide. So we're looking at you take, for example, the sequence of the gene, right? And you have, let's say it's Drosophila melanogaster, and you have a related species like Drosophila simulans, for example. And you're looking at the gene for, I don't know, MyD88, all right? And you're comparing the sequence of melanogaster and simulans, right? If there's incredible purifying selection such that no mutation is beneficial at all, or no mutation that changes the amino acid is beneficial at all, basically what we're going to see is that there can be a lot of synonymous mutations, right, that change the nucleotide changes, but the amino acid does not, right? You can have any number of those. Now, we're not talking about codon bias or selection on codons because that gets complicated. But if we're only tolerating synonymous mutations and we're not tolerating non-synonymous mutations, right, ones that change the amino acid sequence, then our omega statistic here, which is the rate of evolution, is going to be basically zero. Right, we're going to have a really big denominator and a very small numerator. And so as you move closer to zero, it becomes a stronger evidence of purifying selection, right? We're getting rid of anything that is non-synonymous. It's going to change our protein coding, our protein. But if you have a high number of non-synonymous mutations relative to synonymous mutations, that means for whatever reason, it's advantageous to keep messing with this protein. And that's an indication of positive selection, right? We're selecting to change the amino acid. Is that clear?
Is there any mutation-specific bias in those interpretations? Are you assuming equal amount of variation of
This
any of these
assumes
mutations?
equal amounts, right?
Okay.
People have, in population genetics, of course, they've gone back and tweaked these algorithms to say, not every codon is equally fit. And so there's some small selective bias, and even in the technically synonymous mutations based on tRNA, right? But this is just a very simple statistic that you can apply across many different genomes to start to get at whether a gene is under different types of selection.
So then how do you then evaluate this? Because I imagine if you look at organisms, this is happening for many different genes at once, and the fitness is a function of all of them. So like, how do you?
You do this genome-wide, for example, or you take a really big chunk of genes that you want to look at, and you have some kind of reference, just like randomly selected genes, right? So you kind of like relative to a group that you're just pulling randomly from the genome is my group of interest under a different mode of selection. Does that answer your question?
If that's all for coding regions.
For protein coding regions, yeah. There are variants on this if you wanted to look at non-coding regions, but it's a little bit different. Yeah.
But then you're grouping those genes by some classification?
Yes, and I'll
structure
explain
to
that in
then
just a
do
minute.
the-
Yeah.
Okay.
Yeah, sorry, we're running a little bit short.
No, sorry.
Yeah.
Sorry. So we turn to Drosophila melanogaster, and all of these Drosophila species have sequenced and annotated genomes, right? So we can actually look at variation in the sequences of these protein coding genes. Some of what I'm going to focus on melanogaster versus simulans, or looking at the signatures of selection on immune genes in these different species. And so what we can do first is we're like, okay, what are the immune genes in Drosophila, right? People have gone through various methods to try to annotate these, right? There's automatic annotations or computational annotations. There's manual annotations where somebody has actually like knocked out that gene and seen that it has a susceptibility phenotype or something. There's Go terms, right? You can use gene ontology to try to get and so we use a bunch of these different kind of transcriptional, manual, computational annotations to get genes involved in the immune response, genes involved in development, and genes involved in both. All right. And we used 4 different definitions to create kind of variation on this Venn diagram. But basically, we're including the pink circles in our developmental genes. And so we're left with about 250 to 300 genes that we would consider pleiotropic. All right, these are genes that are involved in both immune response and development. Just development, not immune development. And if we actually look at, if we break this down among immune genes into recognition, signaling, and effector, right, most of the pleiotropy is in the signaling class. And this is kind of what we'd expect, right? These signaling modules are being used by multiple traits at different points in time, or sometimes at the same points in time, to achieve the simultaneous expression of traits, right? And so if we actually look at these statistics grouped under these different groups, so we have our non-pleiotropic developmental genes, those genes that we call pleiotropic based on their use in both traits, and then non-pleiotropic immune genes. So these are just used in immune. When we look at that omega statistic, right, the D and DS, you can first of all see there's a lot of variance, right? Some genes are probably under pretty strong positive selection. Most of them seems to be under purifying selection, right? They have a really low DNDS, so non-synonymous mutations are not being tolerated. But if we statistically compare these groups, what we see is that the pleiotropic genes and the developmental genes, the distributions are basically the same. All right, so pleiotropic genes are basically acting like developmental genes in terms of the selective pressures acting on them. Whereas the non-pleiotropic genes actually have a higher DNDS. Alpha is that metric of the proportion of substitutions that are adaptive, right? And we talked about this is kind of a metric of the efficiency of selection. So the higher this value, the less we're tolerating partially deleterious mutations. We're getting rid of them really efficiently. And so you can see our pleiotropic gene class is among the most efficient, right? Any substitution that's adaptive is going to be selected for efficiently. And any substitution that is deleterious is going to be kicked out really efficiently. So these guys are under really strong selection, right? There's a lot of purifying selection. And so how specific is pleiotropic gene expression to one or more stages, right? You can imagine that the more broadly expressed a gene is across life stages, the more that it's a housekeeping gene or it's fundamental to processes of many life stages, we might expect selection to be stronger, purifying selection to be stronger on those because you don't want to mess with them, right? If you mess with them, you're messing with every single process in the organism. Whereas if you have a very specifically expressed gene, right, it's only in adults or only in puberty, then we might expect more relaxed selection on that, right? Because we might be able to tolerate a little more variance. And so we can come up with a statistic to describe the breadth of expression. So if they're broadly expressed, they have a small tau. If they're specifically expressed, they have a large tau. And when we look at the tau, first of all, not surprisingly, right, those that have some kind of developmental function tend to be more broadly expressed. They're working on making an embryo or making a larva or making a pupa. And sometimes they're doing all of those things, right? Whereas the immune non-pleiotropic ones tend to be more specific, and particularly they tend to be expressed in later life stages, right? They're ones that are actually making immune responses. But if we break this, if we take those that are most specific, right? So we're taking all of the ones that are kind of up here, and we say, what's the relative rate of evolution of those that are the non-specific pleiotropic. So we take these and we take the top quartile and the bottom quartile. So the ones that are non-specific, the most broadly expressed ones versus those that are very specific, there's still a lot of variance, but the ones that are specific are under less purifying selection than the ones that are broadly expressed, right? So these guys are just, you can't mess with them. They are, evolutionarily, they're super constrained. And so we actually map those onto some of our signaling pathways, and I won't go through this because we don't have time, but we've come up with some hypotheses for why selection might maintain pleiotropy in these immune systems, even though it's constraining the evolution. And the reason it's a problem that these are constrained, right, is that parasites are trying to mess with them. A lot of parasites would target some of our signaling proteins to try to turn these responses off or to slow them down so the parasites can have more time to grow. And when that happens, right, the host is under selective pressure to adapt, right? And so we should see positive selection in that case. So the fact that these are under strong constraining selection is a problem when parasites are trying to manipulate. I'm almost out of time, so I'm probably going to stop there and take any more questions, but I just want to mention that my grad student, Reese Martin, who's I think a QCD student, has been using some agent-based modeling to actually try to understand why you might maintain pleiotropy in these signaling networks if it's potentially deleterious to immune systems. And it turns out it actually facilitates certain, the evolution of certain qualities like inducible immune responses in our hosts. So I'll skip over this. And I'll just say, right, so the ecology and development ensures that there's not one universally optimal immune strategy, right? You could probably predict that before I told you, but the who, right, is it one parasite type or many? When are you getting infected early or late in life? How, so the actual architecture of those signaling pathways, and then how often? Are you getting exposed frequently to the same parasites, different parasites? Are you almost never getting infected, right? All of these things are going to influence the costs and benefits of mounting and maintaining immune responses. So with that, I'll thank my lab, the people in bold are those who contributed to the work I showed today. A lot of this was funded by GMS, and with that, I'll take questions.
Some students first.
Or maybe two.
I do have a question.
But you.
So in others, so in some species you see gene duplication where you can get, then you can sort of decouple some of these processes. And so I was wondering, how common do you think this is, sort of like using pleiotropy? And then the other side of it is there any ways to essentially tune the direction that this evolution constraint goes into, like any sort of direct evolution ways of trying to improve, I guess, essentially minimize the cost of this constraint?
So first of all, the gene duplication thing, in theory, right? If you could just duplicate these entire signaling modules and then turn one copy into specific for development and one copy, to be specifically for immunity, that seems like it would be a natural solution, but that's not what happens. And so we think, first of all, there's a cost to genome size, right? Like basic kind of selection against like willy-nilly duplicating everything on the earth, because then you have to maintain that network. You have to, keep it around. You have to make those proteins. And so that's one potential explanation for why we don't see this. And like relatedly, having a module that you can use for multiple processes, right? If you can, it's like a Lego block, right? You can pick up that module and plug it into different networks or different kind of detector things at different points in time or in different tissues. You don't necessarily need complete like different Lego blocks if you're using the same one in different contexts, right? So you can imagine there could be different adapters associated with these modules that can modify whether they're acting specifically in immunity or specifically in development. But you still have this problem of selection on the proteins themselves. So it doesn't necessarily alleviate that. There is some evidence for gene duplication in certain circumstances, but that's like aim 3 of Reese's dissertation project. So maybe he'll give you guys a talk in 2 years about the outcome of that, but it is a good question. What was the second part?
Is there any way to like guide this evolution, or essentially direct evolution project? Essentially, is there any way to minimize the cost of this constraint that's being essentially put on these?
Yeah, I mean, so we can do that in silico, and that's what Reese has been doing. When do you, if you start out with a pleiotropy constraint in a module, how does the network evolve around it or something like that? That's what he's done. And it's not that hard to do, it turns out, and over evolutionary time, like a network that started out as pleiotropic and that maintains that constraint can be just as fit as one that was never pleiotropic. It takes longer, but you can do it. And in fact, there's some advantages. The way the network kind of works around that constraint can actually kind of flatten the fitness landscape in some ways and make it easier to achieve different peaks.
Yep. And then my last question was, so you mentioned you right now focused on the protein coding changes, but there's also a lot of cis and trans interactions on regulating the expression, and a lot of these non-coding regions can affect multiple different genes. And so I was wondering, what's your thoughts on taking the whole context of the network in terms of these selection pressures, and sort of whether or not it's more important to change the fitness of a particular mutation of a particular protein to modulate its function versus changing the behavior of when these things are expressed in ways that can affect, instead of modulating one protein, one non-coding region can modulate whole patterns of protein dynamics. I was wondering.
Yeah, I mean, that's one way that you can kind of create these modules, right? Is that what you're asking in terms of like having, do you just change the coding region of this particular protein, or do you change like when it shows up?
Yeah, so essentially this morning, have you looked at the non-coding changes as well? That's what
We have not. No, we haven't looked at that at all yet. It's a little bit harder to look at the evolution of non-coding regions. You can do it, but there's a lot more kind of iffy controls that you need. And I've seen some of these studies published, but I'm not convinced that we have good enough statistical teeth to really do it in our context
And then
yet. So.
how are these, is there a way to experimentally verify these versus like, if I see there's this one gene has particular value, is there a way to then test that to see if it's actually pans out if I ever take these?
Yeah, you could probably, for example, you could insert mutations into some of these genes, right? Or if you know what the parasite is targeting, right? You know, you could screw with it, right? You could use CRISPR to insert mutations or take the sequence of one species and put it into a different species. Because you could leverage the differences in sequences among species. And we have thought about doing that, particularly with Confucium and Castanium for various reasons. We haven't done it yet, but in theory you could.
So when I know you're on the time constraint, but
It is fine.
okay, but anyway, so I wonder if you could comment on the fact that there was an interaction with evolutionary group here. Then artificial intelligence group, an NSF proposal, so maybe. Could you say why? Because there were several proposals, but yours was big, whatever way. Could you say why this is the data that you're using your lab? You are suitable for a machine learning exercise.
Yeah, sure. So it's one thing we're looking at a lot of different axes of variation, right? And we're in terms of like over time, right? We have these time series, and we can get pretty good resolution in many of these time series. We have like RNA-seq data from many time points for individuals, right? We're not looking at the same individual over time because we can't. These are beetles. We can't sample them at multiple time points. But we're capturing individuals, right? And so we have a lot of individual variation. Some of this is coming from some sources, some from others. We don't know why yet, right? So using, if we look at kind of these multivariate analyses of like 16,000 genes or something like that, we may be able to pick up some modules or something where we can explain some of this inter-individual variation or interspecies variation to get at trying to plug it back into kind of our conceptual and like kinetic models of some of these signaling pathways and our regulatory elements. And so if we go back to far back. Right, if we think about like what we're trying to do here, right, this is incredibly iterative, right? We're trying to take how we think the immune system is working, and we can fit these curves to, we can fit these equations to these data to try to parameterize this. But there's going to be some variance, right? And so some of this machine learning and techniques can help us think about where the variance is actually informative versus where it's just noise or something like that. So that could be one thing. And then taking some of this pretty high dimensional data. So this is qPCR data, so it's just what it is. But we have RNA-seq data, right? And we have it over time, and hopefully we're going to have it for many of these different negative regulators, right? So trying to really get at some of these induction and decay dynamics and trying to explain how the slope of this actually kind of goes back to our fitness function, right? Connecting a lot of these kind of high dimension phenotype data or low dimension phenotype data with some of this high dimensional gene expression data would also be a potential avenue.
You mentioned on certain cells and kind of give you an idea of that trajectory of evolution possible to get.
And a lot of this is non-monotonic, right? And so thinking about things that go up and down, the statistics become harder, right, to get at these individual parameters. And we can fit kind of quadratic models and things like that to them, but it's not ideal. So if we can kind of cut through the have more flexible high parameter models where we can appropriately penalize the number of parameters, et cetera, like they do with machine learning, right? So you can also hopefully improve some of our ability to compare these different curves as well.
So one last question. I was curious about your RNA-seq. It's organism-wide, or are you able to actually do cell type differentiation? You're getting
Oh,
transcripts from different cell types?
The data we already have is either gut, fat body, or whole organism. We would like to do some single cell RNA sequencing, particularly for things like the fat body, where there's probably a lot of variance, like particularly when we're trying to explain some of these patterns, right? What's going on here, right? And we think there's probably microenvironments inside the host as well, right? The pathogens are, even though we think about the insects being kind of this bowl of soup because they have an open hemocell, right? That everything's floating around, that we know that's not true, right? They have hemocyte subpopulations that are attached to organs. They have hemocytes that are floating around. Some of them have phagocytose things. Where are these microbes going, right? If we could sample some of these microenvironments and actually get at kind of what's going on, who's actually contributing to some of these signals and the variance to some of these signals. Is that what you're asking?
Yeah, essentially, I was wondering how much resolution you could extract out. I was curious how you were doing that functionally. With the
You
data
could do
got,
that,
so.
right? But I think for some of the questions we're trying to answer right now, it's a little bit overkill to try to do this single cell sequencing, because we're, the power that we want is in the variation across individuals or across species. And so that you can imagine some questions would be more appropriate to think about it.
It's just like kind of as a follow-up, that how diverse is the microbiome like compared to different species? Because I know it's a relative scale, like a human, it's outrageous, but like
It's not. So
Okay, yeah.
in fact, so in our lab, just because of kind of that homogeneous environment of their food, we don't have any
Yeah.
antimicrobial compounds or anything.
right.
They still have almost no microbiome.
Okay.
Like almost, like if we did 16S and we did histology and we tried to find it, nothing. If
Okay.
we go out to the wild and we collect some of these beetles, they do have microbes in their gut, but they're not stable. So if you starve them for a day or two, the microbes disappear.
I see. Okay.
And it's particularly true for the larvae.
Okay.
But part of that is they have this pleiotropy matrix. So when they eat, it's not like it's just going willy-nilly through their gut. They actually encapsulate it in something that makes it hard for non-professional parasites to get out of that,
Yeah.
including just random gut microbes or random microbes from the environment.
Yeah. I mean, for the model, I think that's more ideal. I think.
It's actually nice. It's not this weird microbiome.
Yeah.
Of course, we think about these guys as kind of being part, like we have a lot of protozoa, and most of about 75% of the populations we've captured have these, and of those populations that do, over 50% are infected at any given time with these, which is actually a really high frequency, right? If you think of any given microbiome taxon, it's probably lower.
Yeah.
So can you really understand tribolium without understanding their interactions with bacterias? Who knows?
As someone who works on like more human data, it's interesting to kind of try to accept the space that you're in and feel. So, but yeah, I mean, that's much more ideal to be able to look at parameterize a model. I can't imagine doing that. High dimensional.
Yes,
Yeah.
that's right. Well, the funny thing is we have done some co-infection experiments where we try to, and we infect them with the BT at the same time that we have one or more of these parasites. And co-infection does matter. So it really influences their susceptibility to secondary infections.
That's cool that you have controls and can do.
You can get rid of them.
Yeah, that's great. Yeah.
We don't need special conditions to do that.
Yeah.
All right, let's think. Yeah.